T cells are specialized to recognize foreign antigens as peptide fragments bound to proteins of the major histocompatibility complex. All the effects of T lymphocytes depend upon interactions with target cells containing foreign proteins. Cytotoxic T cells and Th1 cells interact with antigens produced by pathogens that have infected the target cell or that have been ingested by it. Helper T cells, in contrast, recognize and interact with B cells that have bound and internalized foreign antigen by means of their surface immunoglobulin. In all cases, T cells recognize their targets by detecting peptide fragments derived from the foreign proteins after these peptides have been captured by specialized molecules in the host cell and displayed by them at the cell surface. The molecules that display peptide antigen to T cells are membrane glycoproteins encoded in a cluster of genes bearing the cumbersome name major histocompatibility complex, abbreviated to MHC. The human MHC molecules were first discovered as the result of attempts to use skin grafts from donors to repair badly burned pilots and bomb victims during World War II. The patients rejected the grafts, which were recognized as being foreign. It was soon appreciated from studies in mice that rejection was due to an immune response, and eventually genetic experiments using inbred strains of mice showed that rapid rejection of skin grafts is caused by differences in a single genetic region. Because they control the compatibility of these tissue grafts, these genes became known as histocompatibility genes. Later, it was found that several closely linked and highly polymorphic genes specify histocompatibility, which led to the term major histocompatibility complex. The central role of the MHC in antigen recognition by T cells was discovered later still, revealing the true physiologic function of the proteins encoded by the MHC. This in turn led to an explanation for the major effect on tissue compatibility for which they were named. And that is T cells are specialized to recognize foreign antigens as peptide fragments bound to proteins of the major histocompatibility complex. Have a great day.